And now it's time to begin today's agenda. As a reminder, all of our speakers' bios and headshots are available on our One Stop Shop website if you'd like to learn a little bit more about them. But for our first sessions, first we will hear from Ravikanth Kona, who is a pharmaceutical scientist in the Division of Quality Assessment 1 within the Office of Product Quality Assessment, OPQA 1, and the Office of Pharmaceutical Quality, or OPQ, within CDER. Next, we will hear from Reynolds Ray Contave, or Dr. Ray Contave, and Senior Regulatory Health Project Manager in the Enterprise Project Management Staff within the Office of Quality Assurance, or OQA, and OPQ at CDER. And then next, we'll hear from Dr. Paramjit Kaur, a team leader in the Division of Bioequivalence 2 within the Office of Bioequivalence within the Office of Generic Drugs within CDER. So at this time, please join me in welcoming Dr. Ravikanth Kona.
Thank you, Corey, for the introduction. Good morning, everyone. I want to thank you all for being here in person and those connect remotely. My name is Ravikanth Kona. I am a drug product assessor in OPQ. The title of my presentation is General Quality Considerations of Drug Product Label for Alternate Dosing Administration. As the title indicates, I will provide a high-level overview of quality considerations. So what I wish to accomplish from this talk is hopefully by the end of this presentation, I want the audience to take away the following. Understand the general quality considerations of the drug product labeled for alternate dosing administration. Identify the relevant FDA guidances. And learn effective data submission for products labeled for alternate dosing administration. In the outline of talk, I will discuss what is an alternate dosing administration and the common types. I will also discuss quality information needed to support oral label use. Along the way, I will present a few case studies and considerations, and I will finish my talk with challenge questions. So what is an alternate dosing administration? So it refers to a method of delivering a drug or medication through a different route other than the standard or conventional one in order to achieve the desired therapeutic effect. So my talk will primarily focus on these three commonly used use of soft food or liquid as vehicles, enteral feeding tubes, as well as admixtures. So we'll start with the first one, use of liquids and or solid food as vehicles. I want to refer to the draft guidance. It's in the bottom of the slide. It provides recommendation for selection of vehicle and as well as the in vitro test to qualify a vehicle. Image onto your right is taken from the guidance, which lists all different soft foods and their approximate pH ranges. And I think it's critical to understand the pH range of the vehicle that you select because it's going to, for example, if there is an entry-coated tablets or pellets, choosing a appropriate vehicle would impact the drug release. So it's probably going to strip off any entry-coated pellets and thereby altering the pharmacokinetic behavior. I want to specifically refer to the footnote 4 of this guidance. And this is applicable to the generic generic products. So it says the vehicle studies on the RLD would establish the compatibility of the active ingredient and need not be repeated unless there is a risk that the formulation of the ANDA would have a different impact. So in that case, the recommendations provided in this guidance are useful. So what are the general considerations for selection of vehicle? Compatibility and suitability. The guidance recommends that the applicant consider properties of the vehicle such as acidity, alkalinity, chelating or binding effects, especially for entry-coated tablets. pH higher than 5.5 would impact the coating behavior and alter the drug release. Suitability, the applicant is recommended to evaluate flavor, texture, mouthfeel. Any graininess can trigger chewing, again altering the drug release properties. Impact of vehicle, the applicant is recommended to evaluate the impact of vehicle on potency and drug release. Palatability and swallowability, acceptance in terms of taste, aftertaste, smell, texture, swallowability. Patient should be able to take without gagging or choking. In cases for repeat administration or multiple use, it is recommended that applicant evaluate in-use stability data as well as microbial assessment. And in special cases where vehicle mixture is administered along with the feeding tubes, I will discuss the in vitro tests in my subsequent slides. So method of assessing the impact of vehicle on the product quality attributes. Obviously, you need to have a robust analytical method. It should be developed and validated. Any source of interference should be determined if needed. Now, the testing, this guidance discusses evaluating the integrity. Integrity is usually not an issue if you are administering the whole drug product, but if you are manipulating the drug product, say if you are crushing, then any changes to the polymorphism or loss of bead integrity should be assessed. For potency and stability for immediate administration, it is recommended that study be conducted a 2-hour incubation period, and as well as the homogeneity. So I want to point out in the guidance that if the RLD label doesn't indicate the incubation period, the guidance recommends that you perform the studies after 2 hours incubation. Now, for the drug product labels for sprinkles, so sprinkle formulations are drug-containing pellets or granules that can be mixed with soft food before administering. The image I have here on the right side, it shows different ways a drug product can be administered in the sprinkle formulation. It could be in intact, like granules, pellets, powders, or beads, or it could be manipulated, like crushing a tablet, emptying a capsule content, or making serial dilutions, or mixing a syrup into a vehicle. Considerations for sprinkle, obviously the bead size is important. The guidance I've referenced in the bottom of the slide recommends a target bead size of 2.5 mm to a maximum size of 2.8. And bead size is recommended to be performed as per USP 786. And if the applicant choose to provide a bead size greater than the recommended, then adequate justification should be included. Now, there are times where the sprinkles are also administered along with the enteral feeding tube, in which case all the beads are able to safely pass through the feeding tube and not cause any tube occlusions. And the in vitro tests also need to be performed. Now, I want to go on to the case study 1. This is an immediate release capsule formulation. It's a sprinkle. You will notice here the label does not specify the soft food. Also, it's because it's an immediate release. It's an immediate use, so it doesn't specify any incubation period as well. The applicant do not provide any comparability study. Agency issued a deficiency to address the label use. So already performed tests. So these are cellulose-coated pellets. They are not sensitive to pH. They are neutral in taste, so there is no risk for patient compliance. So the applicant evaluated different food here. If you look at the soft food that they have choose, ranges the pH range anywhere from 3 to 8. And as per the guidance, the applicant provided comparability, suitability, impact of the vehicle, palatability, and as well as the incubation up to 2 hours as per the guidance. So the information was found to be adequate. And this is all the testing data that they were provided. Another case study here, you'll see the label indicate the use of 3 different vehicles, cherry syrup, sugar-free or sweet, and simple syrup. And here's the storage conditions. So when the applicant provided the data, the stability data show an unknown impurity when mixed with cherry and simple syrup that was exceeding the ICH limits. The applicant did not provide any qualification data of the unknown impurity, so we showed a deficiency to provide identification and qualification information. So in this case, the API was interacting with the reducing sugars and forming an adduct. So that was showing as an impurity. We'll move on to the enteral feeding tubes. A feeding tube is a medical device used to provide nutrition or medication to patients who cannot obtain a nutrition or medication by swallowing. Based on the site of insertion, we have different enteral feeding tubes. NG tubes, G tubes are the most common, and they vary by tube diameter, tube length, composition, number of ports, connectors, geometry, and eyes. And I have an image on the right side to show the different feeding tubes. So I just want to mention that the variability associated with the insertion point and final sites of drug release are outside the scope of the quality review. So what are the considerations for generic drug products? The applicant should demonstrate the drug product is therapeutically equivalent to RLD when administered according to the label use. Instructions are provided in dosage and administrative section of labeling. And testing information can be found in the RLD packaging insert or product-specific guidance. And testing conditions include number of units, tube type, size, brand, and the size of syringe used. If the label instruction specifies immediate administration after preparation, the agency recommends performing comparative in vitro studies at both 0 and 15 minutes soaking times. Now, what are the in vitro testing? Recovery. So the applicant should evaluate the recovery. It's the percent of the drug of the dose before and after administration. Sedimentation volume and redispersibility. Any insoluble components tend to settle and clog the tube. So the applicant is recommended that they perform sedimentation volume studies and redispersibility. If the solid settles, the applicant should make sure that it can be redispersible before administration. In use study, the applicant should demonstrate that the drug product dispersion is chemically and physically stable. Particle size distribution, acid resistance is in the percent drug release in acid media, and the dissolution. So I want to move on to a next case study. This is an immediate release tablet formulation. Applicant provided data using one material, only 8 French and 12 French. Per the guidance, the FDA recommends the applicant test at least 3 different dental tubing configuration. for the tubes. So we showed a deficiency. The applicant came back and provided the data as follows. And the testing was performed at 0 and 4 hours as per the RLD label. So here's another case study of granules that was building up. In this example, the product-specific guidance recommends the use of NG tube 8 French with 8.5 pH as a worst case. The applicant evaluated as per the guidance. However, a granule buildup was observed and no buildup was seen in RLDs. So we requested the samples. Samples were sent to the FDA lab, which showed large number of granules retained in syringe, and very low recovery was observed in ANDA compared to RLD. And the applicant wanted to, based on the data, applicant wanted to include additional instructions which are different from RLD label, and the product cannot meet the RLD label for alternate administration, which is inadequate measure. So we recommended reformulating it, and the applicant came back, reformulated the product to address the sticking of the pellets inside the syringe. So the next is drug product administered as admixtures. A pharmaceutical admixture consists of a drug product mixed with an appropriate diluent in a suitable delivery device for the purpose of parenteral infusion. ICH Q8R2 section F recommends that applicant perform a comparability or compatibility studies for the reconstitution diluents. Commonly used diluents: saline, 5% dextrose, ringers, lactated ringers. Now here's a case study that I wanted to show where the applicant provided all the information, and we ended up not issuing any deficiency because all the information provided was as adequate. So this one, this product can be reconstituted with water for injection, or also it can be administered intravenously. And the data the applicant provided supports the RLD label use. In raw non-refrigerated water, the applicant provided up to 1 hour at room temperature and 8 hours at refrigerated condition. For IV administration, the applicant reconstituted using sodium chloride and lactated ringer per the RLD label. And the applicant provided appearance, pH, color, clarity, assay, osmolarity, and visible particles. So I want to move on to the challenge questions now. Testing recommendation for in vitro feeding tube study include recovery testing, sedimentation volume and redispersibility testing, particle size distribution study, all of the above. I'll give you a few seconds to think about it. How many of you think it's A, B, C, D? That's right, answer is D. Which of the following statement is not true? Sprinkle formulations are drug product pellets or granules that can be mixed with soft food before administration. Compatibility study with soft food to support RLD label is not a requirement. Agency size of 2.8 mm. Assessment of drug substance in the mixture includes integrity, potency, stability, and homogeneity. How many of you think A? B? That's right, it's B. So in summary, we looked at different drug product quality considerations for dosage form labeled for alternate administration, discussed several case studies along with quality information necessary to ensure the drug product meets RLD label requirements. And hopefully this information provided helps sponsors submit effective data, resulting in fewer review cycles. I want to acknowledge my colleagues at OPQA, Dr. Wu, Dr. Oliver, Dr. Potankar, Dr. Nguyen, and I will also like to thank GDF Planning Committee for giving this opportunity. I will leave with these resources, and I'll be back to answer your questions. Now I'll hand it over to Ray.